Welcome to our exploration of recombinant restriction enzymes, the molecular scissors that transformed modern biotechnology. In this presentation, we'll trace how these proteins emerged from bacterial defense systems and became essential tools for genetic engineering, medical breakthroughs, and our deeper understanding of life, setting the stage for innovations that followed. Chapter 1 opens with the discovery story, and this slide sets the scene for how careful observation and simple lab tools led scientists to uncover a powerful natural mechanism. Think of the microscope image as a reminder that big discoveries often begin with patient study at a small scale. Bacteria turned out to have a clever defense: restriction enzymes that recognize and cut foreign DNA, a finding clarified by Werner Arber's experiments in the 1960s, and ultimately honored with the 1978 Nobel Prize, which recognized how this basic biology reshaped molecular research and opened new possibilities for manipulating DNA. The pioneers behind this breakthrough, Werner Arber, Hamilton Smith, and Daniel Nathans, shared the Nobel Prize for showing how bacterial defense mechanisms could be repurposed as precise tools for genetic research, and their collaborative work laid the conceptual groundwork for recombinant DNA techniques used worldwide today. Bacteria fight viruses in 3 coordinated steps. First, they recognize foreign DNA at specific sequences, then they protect their own genome through methylation marks that prevent self-cutting, and finally they neutralize invaders by cleaving viral DNA before it can replicate, an elegant system that inspired modern molecular tools. Chapter 2 asks a straightforward question: what are restriction enzymes? And invites a closer look at the molecules themselves. This section explains how these proteins act as sequence-specific scissors, why that specificity matters, and how scientists exploit it to read and manipulate genetic code. Restriction enzymes are specialized proteins that recognize short DNA sequences, usually 4 to 8 bases long, and cut the DNA with remarkable accuracy at or near those sites. Thousands have been identified, hundreds are commercially available, and their predictable cutting patterns make them indispensable in labs for creating defined DNA fragments. Not all cuts are the same. Some enzymes create sticky ends with short complementary overhangs that make fragment joining easier, while others produce blunt ends where both strands are cut at the same position, which is less efficient for ligation but useful for particular cloning strategies. So researchers choose enzymes based on experimental needs. To visualize cleavage, imagine 2 outcomes. For sticky ends, the enzyme makes offset cuts on each strand, producing single-stranded overhangs that readily pair with complementary sequences, while blunt end formation results from straight cuts on both strands at the same site, creating clean termini that require different handling in cloning workflows. Chapter 3 focuses on classification and naming, explaining how restriction enzymes are grouped by mechanism and how a standardized naming system lets scientists quickly identify an enzyme's origin and order of discovery, which is essential for clear communication and selecting the right tool for an experiment. Restriction enzymes fall into 4 broad types. Type 1 are multi-subunit complexes that cut far from recognition sites and need ADP. Type 2 are the simplest and most widely used, cutting at or near recognition sequences without ADP. Type 3 have intermediate complexity and different cutting behavior, and Type 4 target modified DNA and are useful for epigenetic studies. The naming convention encodes origin and discovery order. The first letter comes from the bacterial genus, the next 2 from the species, strain identifiers follow, and a Roman numeral or letter indicates the order or variant. For example, EcoRI identifies the first enzyme isolated from Escherichia coli strain RY13, making naming both systematic and informative. Chapter 4 examines how restriction enzymes enabled recombinant DNA technology, transforming theoretical concepts into practical methods for cutting and assembling genetic material and thereby enabling scientists to isolate, clone, and manipulate genes from any organism for research and therapeutic purposes. The cloning workflow is straightforward in 4 main steps. First, restriction enzymes cut both the plasmid vector and target DNA at matching recognition sites. Then compatible sticky ends anneal through base pairing, DNA ligase seals the fragments to create stable recombinant molecules, and the result is an engineered DNA construct ready for isolation and downstream analysis. The cloning process can be seen as molecular assembly. A plasmid and a gene fragment are cut with the same enzyme so their ends match, complementary overhangs find each other and hydrogen bond, and DNA ligase completes the job by forming permanent phosphodiester bonds, producing a recombinant molecule ready for transformation into host cells. Restriction enzymes have had profound real-world impact by enabling genome mapping techniques that helped chart viral and human genomes, powering production of therapeutic proteins like insulin and growth factors through recombinant expression, and providing foundational concepts that later enabled precision gene editors such as CRISPR and TALENs. Chapter 5 turns to factors that affect enzyme activity and how researchers optimize reactions, because getting predictable, specific cuts depends on careful control of experimental conditions rather than simply adding enzyme to DNA and hoping for the best. Optimizing restriction digest centers on 5 key variables: the correct buffer composition with appropriate salts and magnesium ions, strict temperature control around each enzyme's optimum, carefully chosen enzyme concentration to avoid incomplete digestion or unwanted star activity, awareness of star activity risks under suboptimal conditions, and recognition that DNA methylation at recognition bases can block cutting. Looking ahead, the future of restriction enzymes includes continued discovery of novel specificities from microbial diversity, integration into synthetic biology to build complex genetic circuits with precision, and an expanded toolkit that combines classic restriction enzymes with modern editors to offer complementary approaches for manipulating genomes in research and applied settings. In conclusion, restriction enzymes evolved as bacterial immune defenses but became indispensable molecular scissors that unlocked the ability to read, edit, and engineer DNA, sparking a revolution in biology that continues to reshape medicine, agriculture, and biotechnology while providing the conceptual foundation for future innovations.